Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now a part of the Finding Genius Foundation. I have a returning guest. I think this is his fourth time, with good reason. Dominic, he's a research scientist. He's part of the Institute of Human and Machine Cognition. This is all happening down in Florida, University of South Florida, Morsani College. We're going to talk about cancer today because some of Dom's work deals with the metabolic side of cancer. And as I mentioned in other podcasts, I'm putting together a book on cancer. And so I've assembled a big list of questions, and we're going to throw some at Dom and see how he does. So thanks for coming back.
Thanks for having me, Richard.
Before we start, just tell me, in the field of cancer specifically, what's been your lab's approach and what's your just some of your general thoughts about it?
Yeah, well, I was trained traditionally as a neuroscientist, and I did my PhD doing cell biology and electrophysiology and looking at membrane potential and neural networks and things like that. And then as I was throughout my postdoctoral fellowship and academic research career, we developed various tools. And to further, one was atomic force microscopy, which helped to expand the world of nanotechnology. So one of the fun projects that I did as a postdoc is installing an atomic force microscope or an AFM inside a hyperbaric chamber. And to test this equipment, I thought it would be fun to look at different cell types, and how they respond to high pressure oxygen. And, we had the ability to visualize, these cells, and it was kind of the first of its kind microscope. And we had a scanning laser confocal microscope mated to the AFM, to the atomic force microscope. And we were looking at the mitochondria of different cells. And we had a cell line, I believe a U87 glioblastoma cell line. And I saw that the response to high pressure oxygen was much, much different than the other cell types. And I was like, oh, this is interesting, but I didn't really know a lot about cancer. I knew a lot about cell biology and normal cell physiology. I just knew cancer cells were proliferating at very high rates and they did consume more energy, but I didn't really know at the time that energy was glucose. So, I was observing some things, but I didn't know why I was observing that cancer cells were overproducing oxygen-free radicals, and that if I manipulated certain things like glucose concentrations, that, I saw some pretty interesting responses really dictate their rate of proliferation. In particular, I was adding ketones because I was interested in stopping seizures, and the ketogenic diet was for seizures. Okay. And I was actually using I'm kind of embarrassed to say it now because, still a lot of investigators use immortalized cell line. So cancer cells are different than normal cells because they are immortalized. They lengthen their telomeres. Like when they divide, they can keep doing that and just basically live forever, right? So that's why people with, studying longevity will study that. So we have different immortalized cell lines, and the cancer cells are very easy to work with in culture. And this particular cell line was a brain tumor cell line from a 40-year-old, I think, glioblastoma, 44-year-old glioblastoma patient. And I became very, very interested in, studying these cells. And I didn't really understand what I was seeing. So I was reaching out to different cancer biologists saying, why are these cells overproducing high levels of superoxide anion? And I was very interested in how cells produce oxygen-free radicals, because my theory was that this was initiating the seizures that were a limitation of our Navy SEAL divers using a closed circuit rebreather for oxygen toxicity. So it was more of a military. Okay. It was a military project, but I wanted to look at smooth muscle cells and, different types of brain cells. And these cancer cells were just I was just trying to figure them out. And one of the people I reached out to was Thomas Seyfried, because he had done some work with ketogenic diets and had some interesting cancer studies. So he was telling me about the Warburg effect. So the Warburg effect is essentially damaged respiration or oxidative phosphorylation, which normal cells derive about 88% of the energy that a cell makes. And it kind of depends on the cell type. Yeah, go ahead.
Dom, quick question here. Are mitochondria truly damaged in cancer cells where they can't do oxidative phosphorylation, or is it just downregulated and fermentation upregulated?
That question right there is a source of extreme debate between people who sort of embrace the metabolic theory of cancer. I would say that the mainstream cancer community, well, I would say 5 years ago, they would say that the mitochondria are perfectly normal, from a bioenergetic standpoint, meaning that the mitochondria can have no problems making energy, and they are not aberrant or not different in any way, shape, or form. So the main, and I'm not going to name names here, but the people at the top kind of believe that they're mitochondrial damage, that the mitochondria are perfectly fine. And these are literal quotes from papers from top tier, like people associated with the NCI and the top tier cancer. But over the last couple of years, oxygen consumption, because people who study cancer mitochondria use a device called a seahorse. And it basically will give you information about oxygen consumption. So it looks like, from the outside, it looks like cancer cells are consuming oxygen, but you don't know where that oxygen, but it's not coupled to ATP production. And it's difficult to measure ATP production in, using these tools, and you don't know where the ATP is coming from. It could be coming from, we think it's coming from substrate level phosphorylation, not only in the cytosol, but also in the mitochondria. So the Warburg, to summarize the Warburg in one sentence, you would call it, what he observed and what his hypothesis was, his theory was that mitochondrial damage or damaged respiration. And respiration, we think of respiration as breathing in and out, but respiration from a fundamental standpoint is mitochondrial oxidative phosphorylation. Damaged oxidative phosphorylation is replaced by compensatory fermentation in cancer cells, meaning that the majority of ATP, which is the energy currency cancer cells, all cells make, is derived through fermentation, glycolysis, and also substrate level phosphorylation. So Warburg believed that it was the initial damage to the mitochondria. So this damage, there's this oncogenic paradox, right? So many different things can cause cancer: viruses, inflammation, hypoxia, radiation, a wide variety of carcinogenic compounds, age and then also some genetic mutations too. It's interesting. The viruses that cause cancer are the ones that really target the mitochondria. And that's a whole nother sort of topic we can get into. But from the most fundamental point of view, from the metabolic theory, from what Warburg proposed, was that the mitochondria are damaged, and this becomes this is the initiation of cascade of events that leads a normal healthy cell to become a cancer cell. So cancer cells need to maintain negative 56 kilojoules of energy. And it does that, roughly 90% of the energy comes from the mitochondria, and progressive damage to the mitochondria will transition a normal healthy cell to a cancer cell. And I can talk about, go into a little bit more details about that sequence of events. But essentially that's what happens. And in some tissues, it goes relatively rapidly. And in other tissues, there's more of a gradual transition from a normal cell to a cancer cell.
I would think in liquid tumors, the microenvironment of the cancer cells wouldn't be hypoxic or anoxic. But in solid tumors, now you're getting, exterior cancer cells, probably normal environment, middle is hypoxic, and center maybe anoxic. And if you looked across those for their metabolic condition and how they're doing, how they're respirating, maybe then we would get more insight into what's going on the mitochondria level.
Yeah, I mean, you have a pretty good point there. Solid tumors would be, you have carcinomas, sarcomas, and lymphomas. I guess lymphomas you'd put into solid tumors, but leukemia, and that's kind of near and dear to my heart because someone in our family had leukemia. That would be a blood cancer. But the leukemia, it's in the blood, but it starts in a tissue, right? It starts in the bone marrow. So there are different things, and there's a little bit of debate in regards to many of the things that we study are applied to solid tumors. And I know my colleague, Thomas Seyfried, will he had mentioned, and there's a couple papers on this, that in leukemia cells also present a Warburg phenotype, which means they ferment.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
So they may be responsive to these metabolic-based therapies that we are developing and testing. We have not in our lab actually studied any of these blood cancers. Although I'm of the opinion that metabolic-based therapies may have some utility in the management of these blood cancers. But I mean, from a purely fundamental perspective, I think there are we know that certain forms of leukemia can be triggered by carcinogenic agents. So, and these carcinogenic agents are known to damage the mitochondria, and which creates an energetic crisis, which then would in some way sort of destabilize the nuclear genome of the cells, right? So the bioenergetic state of the cell really dictates DNA repair mechanisms. And if there's an energetic crisis, the fidelity of the nuclear genome will be compromised. And that can trigger, oncogenic activation. Things like HIF-1 alpha, tumor suppressor genes will be compromised. And then you have a gradual transition from a cell that uses oxidative phosphorylation to compensatory fermentation and substrate level phosphorylation. It's almost a de-differentiating back to a more archaic form of energy production. So the mitochondria are really like a separate organelle or a separate organism, almost, if you want to take it that far, that like live inside the cell, right? And they have their own DNA. I think there's 13 different genes involved in it, and they have their own DNA repair mechanisms. Although the DNA repair mechanisms of mitochondria are less robust because there's a lot of them, right? And you have mitochondrial biogenesis and things like that going on. So maybe not as it can take a hit, but it can only take basically all the agents. So my point is that all the agents that cause cancer. Radiation, hypoxia, inflammation, viruses, the aging process, various carcinogenic agents, they have a greater effect on the mitochondria than they do directly on the nucleus, nuclear DNA. Even radiation, if we want, I'm actually doing a big review now on space radiation. So it's called galactic cosmic radiation or solar particle events, these high, charged ions that we get bombarded with when we go into space, because we do a lot, different projects with NASA and things like that. So kind of really delving into that and come to find out radiation, if we're just going to focus, radiation is kind of an awesome thing to study if you're going to study cancer, because it's such a powerful carcinogenic agent, if you want to call it an agent or stimulus. And if you cause it can directly cause double-stranded nicks in the DNA, which could cause an oncogenic transformation. But what radiation really does is damage the mitochondria in a way that will cause inflammation, and that inflammation can further damage the mitochondria, and it really produces an energetic crisis, and that through what you want to call the retrograde response. So there's an intimate communication between the mitochondria, crosstalk, but the mitochondria is always talking to the nucleus. And, and when it's telling the nucleus, we have an energetic crisis and we cannot make enough ATP via oxidative phosphorylation, there will be oncogenic activation of oncogenic pathways or oncogenes that basically allow the cell to survive, things like HIF-1 alpha and VEGF and things like that. And these oncogenes will increase glycolysis, will increase various growth factors that will start to initiate that transformation of a normal healthy cell to a cancer cell, which will express all the hallmarks of cancer ultimately, which is evading growth signals.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Limitless replicative potential, anti-growth signals, sustained angiogenesis. Cancer cells can also evade apoptosis, right? And then there's metastasis, right? Invasiveness and metastasis. Only, I think it was the Hallmarks of Cancer paper was written, kind of forget now, but around 2000, yeah. And then 2011, they finally added, only after, some criticisms of this did they add the Warburg effect, which is, energetic, bioenergetic damage or transformation and changes in the energetic mechanisms.
But this is a different one. Are there epigenetic changes in the mitochondria only? Has that been observed? And perhaps that's an early changeover in the cell versus nuclear epigenetic changes?
There is. And I have a student, looking at a genetic disease, and we're actually looking at ketones as an epigenetic modifier through something called beta-hydroxybutyrylation. So beta-hydroxybutyrate, which is a ketone, can directly interact with the histones. From a mitochondrial, so you're asking about mitochondrial epigenetic. Well,
here's why I asked. If there is mitochondrial-based epigenetic changes, perhaps it downregulates OXPHOS and upregulates fermentation to a degree where, to an outside observer, it looks like the mitochondria is damaged and can no longer do OXPHOS, but in fact, maybe it's just a silencing of it and a upregulation of this alternative pathway.
Yeah, there's a dynamic interplay there that I don't know that, and it may vary between different tissue types and cell types and things like that. But there's no doubt in my mind that there are a wide variety of epigenetic mechanisms occurring probably at the level of the mitochondria more than the nucleus. We do believe that the mitochondria kind of call the shots. Like if you have robust and healthy mitochondria, then you generally have DNA repair mechanisms that are sort of maintaining, the fidelity of the nuclear genome, and repair processes are going on as planned. Those repair processes are very, very energy dependent. So a decrease in the bioenergetic capacity of the cell will have a tremendous magnifying effect on the DNA repair mechanisms. So that's why, this further sort of solidifies the metabolic theory in my mind. But the thing is that we can be susceptible to this in a wide variety of ways. We can have various SNPs, and, I spent quite a bit of time studying redox control, antioxidant mechanisms, and, the immune system also comes into play. So it's probably more complicated than anybody can. My colleague, Tom Seyfried, thinks that, cancer is very simple. And if you view it from a metabolic standpoint, it does become a lot more simple. But I also think you have to factor in, the immune system. You have to factor in a lot of the epigenetic regulatory events that I frankly don't know enough about. But I do know that, metabolic controlled epigenetic regulation is dictated by the health and vitality of the mitochondria. So I can go that far and say that. And we are looking at various mechanisms, but when I start and put all my time and effort into studying a few mechanisms, then, a dozen more come out, but we try to sort of focus on the low hanging fruit and the mechanisms that have the most robust control, that are sort of this interplay between metabolism and epigenetics is a big focus of what we're doing now.
Quick question here. What organisms do you know of get cancer and which ones don't? And what does that tell you? And does this go down to single cell organisms? Do they ever get cancer?
Oh, wow. That's a good question. No one ever asked me these questions. These were really good questions. I have to be honest. I haven't really given this a whole lot of thought. I'll just tell you what's coming to my mind. I know plants, for example, I'm looking out my window now and I see some trees that actually have tumors on them. So these, trees, plants can get tumors, and I I'd have to look into this, but I don't think you'd call them cancer. I know insects, get malformations that sometimes people cause, cancer. And even like mosquitoes, for example, I had a friend studying mosquitoes and he's saying, oh, he's seeing cancer. He's seeing these malformations. But when I think of cancer, so you can have a tumor that's benign. You can have a tumor that is pre-malignant, or you can have a tumor that's malignant. When I think of cancer, I think of invasiveness and metastasis. And I actually should probably, really, the definition of cancer is kind of fuzzy to me. And I don't know if it can be applied to your question, which would be a single cell organism. So I don't, to my knowledge, and maybe someone could prove me wrong, I don't think a single cell organism can get cancer. I know when you go up the chain, obviously we studied mice and they get all sorts of cancers. Most of them will die of some form of a cancer. But when you work your way back to like insects and then smaller, multi-cell organisms, you start to see growth and things that would resemble cancer. But from a mammalian point of view, the definition of cancer is usually when we call it something that would kill the organism. So like a malignant cancer. But you can have different cancers that are benign too, right? You can have like a benign tumor, but I think some people would not maybe label that a cancer because it doesn't have the capacity. Most people don't die of a tumor, right? They die of tumor burden once that tumor becomes metastatic and once it invades other tissues and impacts the function of those tissues and spreads. But maybe it's a long-winded question, but I don't know, to be honest, I don't think single-cell organisms can get cancer, but I know multi-cell organisms get growth and malformations that resemble cancer. So that's my best answer.
And then, also other forms of life. Do bacteria ever get anything that, looks like this, or a biofilm, or, does cancer manifest in protists or, non-eukaryotic cells?
Well, yeah. So again, I don't think you would label that sort of malignant cancer, which would, although some growth things can, kill plant, for example, and different organisms, but it differs in that, the cancers associated with mammals are associated with invasiveness and metastasis. And I think you can have benign cancers like prostate cancer, for example. I'm trying to think of indolent cancers. So I think we would label them indolent cancers where people, and this is actually a very hot topic right now, actually over-treating people. So you can do a scan and then detect cancer, like prostate cancer and various indolent cancers. But if you go in there with chemotherapy and radiation and stuff, then you're basically giving someone a very pro-carcinogenic therapy. The person is coming out of that therapy at a greater risk of even different forms of cancer now, especially if you impact the immune system. So this brings up the idea, that are humans living with cancer? So, there's many people who die and upon autopsy, you find out that they have tumors in them and they were perfectly fine throughout, and who knows how long that they had those tumors.
Yeah, is there any program to, as part of autopsy, it wouldn't be like an organ donation, but I guess it'd be like a histological, pathological donation to look at and sequence, what appear to be non-lethal tumors in older people that have died. They're there, but they don't seem to have metastasized or caused disease, and, to sequence them and look at them.
Yeah. I'm not sure how much information that you'd kind of garner from a deep analysis of these. So what we have observed is that the more the mitochondria are damaged, and by mitochondria, I'm talking about you have structure, number, and function, right? So my colleague, Tom Seyfried, and others now are kind of coming on board with this idea that in all cancer cells, maybe even leukemia too. I think leukemia may apply to this. So no cancer has normal mitochondria. I know not everyone would agree with this. I mean, 10 years ago, the large majority of the cancer biologists would say that's absolutely not true. But I think they're coming on board. And now you probably have about a third of them will say that's true, maybe even half. So, and I remember Tom telling me more than 10 years ago, it's just a matter of time. Because I was telling him, I'm reading this paper, I'm reading that paper, and these great researchers, and I admire the research, are telling me that, cancer has normal mitochondria. And, he was just saying it's just a matter of time. And I've kind of seen that shift over the years. So when it comes to indolent cancers or benign cancers, the difference is that those cancer cells, they ferment, but their mitochondria are only damaged slightly. So they have less mitochondria. There's mitochondrial abnormalities, and functional abnormalities would be a decrease in a lipid called cardiolipin. So, studies have been done looking at there's a microbiome and there's there's the lipidome, right? The lipidome is sort of like a global analysis of the lipids in the mitochondria. And what you see is sort of an immature cardiolipin. The cardiolipin is kind of the glue that holds the inner mitochondrial membrane together. And if you have immature or less cardiolipin, then the mitochondria literally cannot produce ATP. That's kind of like the business end of the mitochondria, the inner mitochondrial membrane. There's a decrease in the sort of the structure of that membrane. And as that progressively decreases, then the mitochondria fail to be able to make energy, and then there's a transition towards glycolysis and substrate level phosphorylation. So there's data that's accumulating that this substrate level phosphorylation, which we thought was only in the cytosol, is actually occurring in the mitochondria now. So the mitochondria can actually start making ATP through substrate level phosphorylation. So anyway, the point I think I want to kind of circle back, that these indolent benign cancers do not have that massive, mitochondrial damage, structure, function, number that you find in more aggressive cancers. So stated another way, a hallmark characteristic of cancers that are highly aggressive and highly prolific and highly metastatic is that they have a higher degree of mitochondrial damage, defined as structure, number, and function is aberrant. So you'll see less mitochondria. You'll see, functionally they're different, and then they have their biochemical signature will reflect a greater or less cardiolipin, mature cardiolipin. So they'll have a proportion of more immature cardiolipin, which is a component of the inner mitochondrial membrane. And these are just hallmark characteristics of the mitochondria of tumor cells that are highly metastatic and aggressive. And the tumors will have a very pronounced Warburg phenotype, which means that they are fermenting at very high rates, levels of glycolysis that are, reported to be 100 or 200 times more. So that the cancer is very, very hungry for glucose and glutamine, which is a little bit harder to target. But so we're very interested in sort of understanding these processes and understanding the metabolic phenotype of cancer. And we feel that, there's immune oncologists, there's radiation oncologists, there's, oncologists that, deliver chemo. There needs to be a whole new class of oncologists called metabolic oncologists. So we are
functional, yeah.
Yeah, and we're understanding that these metabolic therapies, and it doesn't, diet is one that should kind of be the cornerstone, but there's a whole toolbox of, metabolic drugs that are being developed that are far less damaging to the patient's long-term health than these chemotherapy drugs, which wipe out your immune system, which makes you susceptible to other forms of cancer. And they may give you a couple extra months, but they don't increase your chances long-term surviving. That's what.
Yeah, you may have been involved in this or know about it, but what happens to the respiration under hyperbaric oxygen, high pressure oxygen? That would turn hypoxic, anoxic zones into more oxygen rich. Does that do anything to the mitochondria? And if so, maybe that means it's not a permanent damage to them, but again, just a change in regulation.
Yeah, well, that's interesting. So that was sort of what got me into this whole field of cancer. When I was a neuroscientist, studying neurons, and we threw those brain cancer cells in. Although neurons generally don't transition into cancer cells, the glia do. So the glia just have a higher capacity, higher susceptibility, oncogenic transformation, and the microglia too. So, but that's another story. So, when studying these brain cancer cells, what we observed is that under high levels of oxygen, they overproduce massive amounts of superoxide anion. Like it was literally just like maxing out the fluorescent signal that I was measuring. So I had spent a lot of my postdoctoral fellowship developing techniques to measure superoxide anion in hippocampal brain slice preparations. So using different tools and techniques and dyes and things like that. So when I threw these cancer cells in there and exposed them to hyperbaric oxygen, it like saturated the signal. And I had actually used less concentration of the dyes. So it's just like I actually had to come up with a different technique because the cancer cells were just blowing out massive amounts of lactate, presumably, but also blowing out massive amounts of oxygen-free radicals, these reactive oxygen species. So I was using a dye that measured superoxide anion. So we know that as a tumor grows, as the biomass of the tumor grows, it outstrips its ability to the blood vessels don't keep up. And as the cells divide and the tumor grows, the blood vessels can't supply enough energy, nutrients, and oxygen to the center of the tumor. So that the center of the tumor becomes hypoxic. The hypoxia inside the mass of a growing tumor further damages the mitochondria and oxidative phosphorylation. And you get, inflammation, and then the tumor is producing these reactive oxygen species, and then the reactive oxygen species, further damages the nuclear DNA and the mitochondrial DNA. And so the idea that, you just asked me about is that with hyperbaric oxygen, when you deliver hyperbaric oxygen to a person, that hemoglobin is saturated, but what you're doing is you're putting oxygen into the plasma, into the liquid part of the blood, right? And because the blood vessels are sort of erratic and compromised, the red blood cells kind of get caught into little pockets into the tumor, and the red blood cells literally can't get inside the tumor. But if the plasma is saturated with oxygen, and you can saturate the level of oxygen in the plasma by like 2,000%. The PO2, if you just stick a PO2 electrode inside of a solid tumor and then give someone hyperbaric oxygen therapy, the levels of oxygen will go up like 2,000%, like crazy. What you're doing is not only are you reversing tumor hypoxia, which could help silence some of the oncogenes and also increase the tumor suppressor gene activation, which is beneficial, and those studies have been done. Most importantly, what you are doing is actually you're causing an oxidative burst inside the cancer itself. So the cancer cells will proportionally overproduce oxygen-free radicals relative to the healthy tissue surrounding it. So you can sort of more or less get a site-specific overproduction of oxygen-free radicals. And this can be important, especially in the context of other therapies. For example, chemotherapies that work through an oxidative stress mechanism can further be augmented by hyperbaric oxygen. Also, the efficacy of radiation therapy is directly proportional to the PO2 of the tumor. So if you elevate the partial pressure of oxygen inside the tumor, that will further sensitize that tumor to radiation therapy. And this could be, can you imagine giving someone hyperbaric oxygen and doing a targeted radiation procedure of that tumor? You could use far less radiation. You could probably use 5% of the radiation. And I'm not even exaggerating here, and if it's targeted with a gamma knife or other, seeds or things like that, then you're essentially doing no damage, very little, if any, damage to the healthy tissue. But because hyperbaric oxygen site-specifically causes a massive overproduction of oxygen-free radicals, which is the overproduction is directly proportional to the Warburg effect, right? So with the Warburg effect, you have a more hypoxic tumor. We're talking about a solid tumor scenario. So the more aggressive that solid tumor, the more it's going to be responsive to a hyperbaric oxygen therapy that's done in tandem. Alternatively, if you give someone hyperbaric oxygen and you take that person out of the chamber, the reactive oxygen species inside the tumor will stay elevated for an hour or two after. We know this. So you could potentially give someone hyperbaric oxygen therapy and then put them into radiation therapy, or give them radiation therapy and then transition them, just walk them over to the next door into a hyperbaric oxygen chamber. And many hospitals have hyperbaric oxygen chambers for wound healing. So there's 14 different FDA approved applications for hyperbaric oxygen therapy, and diabetic wounds or ischemic wounds is one, carbon monoxide poisoning there's actually radiation necrosis. So if someone is actually getting radiation therapy, their insurance would cover the use of hyperbaric oxygen therapy if they're already getting radiation, because it's an FDA approved application.
I wonder, as a poor man's therapy, you could use the Wim Hof breathing if you're going to get radiation, and perhaps that would have some of an effect.
It's interesting you bring that up, because there's definitely something to that needs to be studied. Right. So we could potentially increase the level of carbon dioxide. So carbon dioxide is a very powerful vasodilator, and we can manipulate our physiology in a way that could hyperperfuse the tumor tissue in a way that could really enhance other forms of therapy. It involves a lot more time and effort and maybe expertise too. A lot more time and effort and nuance for the patient. And every patient may not be able to do it, but I do think that these breathing techniques have a lot to offer to cancer patients in further augmenting the therapeutic efficacy of different modalities.
I know there's many mechanisms, but what are some of the mechanisms that cause cancer to start from inception, and what are some mechanisms that you hypothesize would start cancer in somebody?
Yeah, so this is kind of like the oncogenic paradox, right? There's so many different things that can initiate cancer. And we believe that many of these things that initiate cancer directly cause an insult to oxidative phosphorylation to the mitochondria. So, people listening to this want to know, what can they do to prevent these initiators of cancer? There are different viruses. So maybe we can kind of focus, I'm not a virus expert, but I do know that, things like there's DNA and RNA viruses, both have been implicated in the etiology of cancer. And that could be the HPV virus, the herpes virus, the Epstein-Barr virus, hepatitis viruses, the HIV, Kaposi sarcoma, for example, for HIV. So these viruses, and that was one of the things that, I first asked Tom Seifert, what about viruses? Viruses cause cancer by directly impacting the DNA and causing oncogenic activation. But he assured me that the viruses were targeting the mitochondria. And, if you actually start to do a deep research, especially some of the new research that's coming out, further suggests that these viruses are really compromising the mitochondria or causing inflammation, which then damages the mitochondria. So, I actually had a shingles episode like a year or two ago. Maybe it surfaced, but if I fast, I noticed that fasting quickly silences the shingles. I was under stress at certain points in time. So, and I think maybe that could be a virus too. So I think that viruses, if you have various viruses, I think there are various antiviral agents that can help. Aging, the process of aging itself, you have a decrease in mitochondrial capacity and mitochondrial health as you age. So things like, of course, diet, fasting, exercise is the big one, breathing, meditation, all these things can help increase, our respiration and enhance, our mitochondria. But the mitochondria are really the ultimate tumor suppressor. And I want to emphasize that if you have healthy and robust mitochondria, and then some people worry about things like EMF and radiation from devices and things like that. I get asked so many questions. So I'm actually bringing that up now. What about the cell phone? What about EMF? I think these things are negligible. I think you could strap a whole bunch of cell phones to your head and, I don't think it's going to increase your chances of brain cancer. But I do think that the interaction with the environment and the immune system, there are various carcinogenic agents that we probably need to pay attention to. I live on a farm and we're kind of phasing out. We want to avoid using any pesticides, herbicides, things that can permeate and get to the food system. And genetic mutations really account for only 5, maybe 6% of cancers that we know of. And many of these genetic mutations, which people will say affect the nuclear genome, many of the genetic mutations actually target the mitochondria. But I think if we're talking about cancer prevention, which I think is, for the listeners out there, what you want to do is basically have robust metabolic health through some amount of anaerobic exercise. I'm a big believer in strength training. Your skeletal muscle mass will dictate your mitochondrial health, because if you build strong and healthy muscles, you make your body very hungry for glucose, you decrease inflammation, you increase your lifespan, if you are metabolically fit in terms of strong health. And what I like to do is use a cardiometabolic kit. So there's a company called ZRT Labs. And instead of having to go to Quest Labs or LabCorp, I get a kit. I go to Amazon.com. I buy the ZRT cardiometabolic kit, and it measures HSC reactive protein. It measures insulin. It measures hemoglobin A1C. It measures, triglycerides and all this stuff. And I don't have to go get a prescription. I just, I go online and I buy this thing. And then I squeeze a couple drops of blood onto a card. I seal it up. I send it to the lab, and they email me the results. I do my hormones that way. I actually go and get, maybe once or twice a year, I get a CBC, complete metabolic panel, or a CBC and a CMP. So complete blood count and a complete, metabolic panel. But the important ones that people want to focus on are your blood glucose control. So I wear a continuous blood glucose monitor, and I use Levels Health software system. And it's a program, and you can sign up through Levels Health, where you wear a CGM device on my arm, and it goes to, I'm using the Abbott Libre, but it's also working with Dexcom, and goes right to my cell phone. And I can look at my glucose response to meals, to what my glucose was doing when I was sleeping, and I can evaluate my nutrition. So if I eat something that spikes my glucose, I know not to eat that or to eat less of it. And then over time, I can train myself on the best way to eat to manage my glycemic variability. So glycemic variability and a cardiometabolic kit are probably the most important things that you should focus on to optimize your metabolic health. And metabolic health will basically equal tumor or cancer suppression, or cancer prevention.
I know this may be a little esoteric, but the biogenesis of mitochondria versus the biogenesis of ribosomes or other cellular structures, is it different? Do mitochondria, for instance, only come from other mitochondria? Do they seem to retain their sense of self, their separateness from the rest of the cellular structures, or are they similar to, again, the biogenesis of other organelles?
Yeah, they tend to, it's a good question. And a lot of people are studying this, not our lab in particular, but they tend to sort of bud off from one another, and they can, proliferate. The mitochondrial biogenesis will be dictated by the pressure that you put on the metabolic system. So meaning that, if you're eating a high carbohydrate diet and you're sustaining on that, and then you transition to a high fat diet or you start fasting, then you move the energy system, the demands, which would be, you move away from glycolysis and you move towards a reliance of energy on oxidative phosphorylation. And for your body to keep up with that energy demand through the OXPHOS system, it puts a stress on the mitochondria. So in the beginning, there's actually an increase in reactive oxygen species and things like that look negative. But these are part of the adaptive response. And you have a number of different pathways are activated in part from the nuclear to mitochondria retrograde response. So the mitochondria signal to the nucleus sort of in energetic crisis. So, and in the context of fasting, it's a favorable adaptation where there's an increase in the function, in the structure. You have structural changes, and then the number of the mitochondria will adapt over time. And, different intermittent fasting, fasting itself, and a diet, a ketogenic diet can actually increase mitochondrial biogenesis. And the ribosomes that's another question I'm not knowledgeable enough to ask, but I know when there's an increase in protein turnover and, the demand for the body for growth and repair, there's sort of an upregulation and expansion of the ribosome organelle to keep up with the demands of that.
Okay.
It's a dynamic system, so it can change, decrease and increase depending on the demands on the organism.
Well, the reason why I ask is I wonder how, I know that mitochondria resemble, I guess, a certain kind of blue-green bacteria or blue-green algae. They seem to be kind of a microbiome within the cell. So that's why I wondered if the biogenesis is different and, mitochondria only begat from other mitochondria and the other cellular structures are, kind of created by the entirety of the cell itself, if, how separate they are. And it makes me wonder if cancer, I wonder if you could say it's really a disease of the mitochondria and not of the cell itself, not of the whole cell. And it's the interaction of the mitochondria with the cell that really determines the nature and extent of cancer.
Yeah, that is a good question. And I think, sometimes questions pop into my mind that I write a note to go investigate. And the link between your susceptibility of getting cancer and your sort of maternal genetics. So looking at like your mom's background. So we inherit our mitochondria from our mothers, right? So, I believe that sort of our susceptibility to cancer may be linked. There's a greater link from the maternal side because we get our mitochondria from the maternal side. So I haven't looked into that, but I think someone has to have looked into that. But I think that's an important thing to investigate or to look into.
All the abilities and the hallmarks of cancer, I know that they're postulated in this paper in 2000. But what do you think the order of appearance of these mutations or abilities is in cancer? When it first starts, what do you think is the first ability, and then the next, and the next? And I know cancer and tumors are very heterogeneous, but is there any indication by looking back through lineages, within a tumor, from metastasis to primary, can you see like what came first, what came next in terms of ability?
To the cancer biology class as an invited speaker, we kind of hit on this. So it does vary between, the type where the cells are, for example, whether it's a liver, or kidney, or brain, or whatever. But if you go back to the hallmarks of cancer, which I think you're kind of referring back to, I have to say that it's the initial insult that will transform a normal cell to a cancer cell would be that energetic crisis and then the oncogenic activation, which really, I guess the first sort of thing that you'll see, if that's the sort of the question, like what do you see, is sort of a self-sufficiency, like the cell kind of de-differentiating. And this is sort of my interpretation of the literature. There's a transition to the default de-differentiated state where the cell sort of just wants to become autonomous, right? So it goes back to this like archaic form where it becomes self-sufficient in growth signals and it becomes insensitive to anti-growth signals. And this endows the cell with transition to proliferation, ultimately unbridled proliferation. But I think the first thing and I think if I'm understanding your question correctly, it's the first thing that you start to see with oncogenic transformation is a self-sufficiency in growth signals. So it goes to a default state of de-differentiation. And then it becomes like its own organism, like it's separate, almost separate from the body. It walls off itself. It creates various proteins and glycoproteins, and then it becomes, it becomes sort of resistant to the immune system recognizing it. Usually natural killer cells will recognize it, and there's various, cell surface markers. But as the cell starts to proliferate, it starts pumping out lactate because it will present with a Warburg phenotype. And that also contributes to the cell's, the cancer's ability to evade the immune system. As it starts growing and starts fermenting and it starts pumping out lactate, and that changes the microenvironment of the tumor, and then the immune system can't recognize it. But I think that the first sort of signs are the cell actually becomes autonomous. It's like a kid that like becomes sort of, defiant in the family. It's like it wants to avoid everything and just wants to completely wall itself off and be its own independent entity. And that process is a consequence of an oncogenic activation, an oncogenic program, if you will.
And maybe you could say metastases are like runaway cancer cells. They ran away from home.
Yeah, yeah, I think of metastasis and invasiveness are sort of further down the line. It's like, I don't know. Maybe I'll put this into like an analogy or whatever. It's like you have a kid and it's just like, you have a very toxic environment in the family. And this is just a stretch here, but the kid like closes himself in the room and wants to become an independent entity and, is totally ignorant of any kind of signals that the family is giving. And, maybe he has a group of friends and he just starts, replicating his ideology with them. And then he goes off and starts, propagating his negativity and forming, goes into a gang, right? And that's really what a gang is. If someone, you want to put it in that kind of analogy.
If cells go self-sufficient and they're autonomous, at what point, if you have quote unquote enough of them, now do they act in concert? Do all the cells of a tumor, even though they're heterogeneous, act in concert to further that new organism that is cancer? Does it act as one? Does it have a sense of self and act that way at some critical mass?
Yeah, well, that's a good question. So, it kind of depends on the primary location of where the cell is. And then, as that cell divides and proliferates and then becomes endowed with invasiveness and metastatic potential, then it gets in the bloodstream and then it ends up in like the liver and the kidneys and maybe in the brain. And, it has those same qualities, but it is very genetically heterogeneous and can start to transform in different ways and become more aggressive. There's evidence too that with metabolic therapies that an aggressive tumor can actually become less aggressive unless it can actually decrease in grade and in invasiveness over time. But it does, there is, I think what you're getting to is, does it like communicate with the original primary mass? It does definitely have some qualities. And I think, there's probably vesicles and RNA that's being, in the bloodstream that's starting to influence other tissues surrounding it. I think you had questions about the microbiome. I know that tumors in a particular organism, in a particular organ, like if it's in the colon, if it's in the liver or different areas of the body, can change the whole microbiome of that particular organ. And that microbiome can then have epigenetic effects. So it's like the tumor, by virtue of changing the microbiome, and the microbiome is responsible for making everything from, serotonin to, a whole cascade of things, and including, different inflammatory factors too, that can influence growth and invasiveness. So by virtue of not only the expanding biomass of the tumor, but also influencing the microenvironment and the microbiome of the particular organ, can then produce epigenetic changes that can further alter the whole metastatic cascade and maybe further influence and could be a way that primary tumor is talking to other tumor cells throughout the body. So, this is kind of getting far out there. This is like on the fringe of what people are studying now. But I know that quite a few papers have come out on how the tumor is changing the microbiome of different organs.
Well, I guess to boil it down, what I mean is this. If I have, from what I understand, a 1 centimeter sphere of cells is about 1 billion cells, and that's like close to the smallest visible size of tumor. So if you have 1 billion cancer cells, yes, they're heterogeneous, but are they acting as one creature? Are they acting in concert? Are they signaling to each other? And they're all saying, all right, in part of us, we need some angiogenesis going. There's an anoxic region growing. Do they act essentially like a biofilm, or do they act like a tissue or an organ, or are they just kind of a bunch of mercenaries that are together, but they don't really work in concert.
They secrete growth factors, they pump out lactate, they change the microenvironment. They are coupled through even like neurotransmitters. They are coupled through things called gap junctions. So gap junctions are sort of, they create like these hemichannels, and there's connexins that form a hemichannel. And through these connections, they can be electrically coupled, and they can also be metabolically coupled. So if you have a tumor, you could go on one side of the tumor and cause like, a biochemical change in that cell can happen. And through various gap junctions that tumor is sort of networked with the other cells in the tumor. So I know that that's the way it works in physiological systems, that have network properties. And I do think of tumors as having sort of these network properties. So they are all communicating to one another to maximize growth and proliferation. But the process too is also a bit erratic. And I say it's erratic because a hallmark characteristic of one of the hallmarks of cancer is actually sustained angiogenesis. That sustained angiogenesis in a solid tumor is very erratic. And which means like if you look at the blood vessels of the tumor, it's not like they're going everywhere. It's kind of like a disorganized, confused individual who's just like kind of schizophrenic and erratic. Like it's not a very elegant, coordinated sequence of signals that would cause the blood vessels to grow to maximize the growth of the tumor. And that always kind of, I was always a little bit like confused about that, and that you do. And that's actually why hyperbaric oxygen is so effective at reversing tumor hypoxia, because the expanding biomass of the tumor makes the core of that tumor anoxic because the blood vessels are not coordinated to grow. So the cells aren't talking to one another in a way that happens in normal cells and normal tissues, in a way to make those blood vessels grow in a very, elegant and defined fashion.
So it's like cancer's like a bull in a china shop. It's still an organism, a bull, but it's going to wreck the china shop, meaning it's the surrounding body. It sounds like that. That's my analogy.
Yeah. And I do think, the more I think about this now, and some of these things I haven't really thought about in depth until you just asked them now. So I do think that, kind of cells are much more autonomous. So they're not just a unique metabolic entity. I'm not saying like each cell is like that, but each cell is autonomous in that it's not, the growth factors and signals and things like that are much more erratic than what you find in normal healthy tissues, because you have cascades of events that are turned on that are just not coordinated in a way. And this gets into like this question is like, what is the evolutionary advantage of having cancer? I mean, these are like deep questions. I remember, I think it was Paul Davies that at University in Arizona that invited us to, and Tom Seaford was there too. And we had this roundtable discussion, philosophical discussion, essentially. It's like, what is cancer? And it was like a meeting of the minds. And I was like actually shocked that I was invited to it, but I think I was kind of a new guy studying this, and they wanted some fresh perspectives, but there was definitely some high power people there that were just talking philosophically about some of these questions.
Okay. Well, very good. Where can people find out more about your work and everything you're doing in your research papers?
Yeah. So, one-stop shop would be Ketonutrition.org, Ketonutrition.org. And we have a blog there, and we post, couple blogs a month, and we also have a newsletter. So if you go to ketonutrition.org, sign up for the newsletter, and you'll find all the information on our latest publications. We evaluate different studies that come out. If a product is really interesting to us, we put it in our newsletter. When this podcast comes out, I'll put it in the newsletter and spread it out through that medium. But ketonutrition.org.
Very good. And Dom, thanks for coming. Like I said, I've had you 4 times. I hope to have you a bunch more in the future. So thank you.
Well, I appreciate being on. Thanks for having me, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at FindingGeniusPodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.